Yeah, wherever you are, I wish you a good morning and welcome to the lecture Applied Microbiology. Yeah, our first lecture, it's about the introduction, and of course, I also want to talk to you how these things will look like in this semester with online teaching and so on. Let's start with this lecture. As you see, this lecture will be a YouTube live stream, and hopefully most of you are sitting here. And maybe I first explain why I decided to do it in this format, because there are, of course, many options to do the teaching in a pure digital manner. First of all, maybe for you, as I'm your head of study course, just to remember the optimal thing, our university is tailored to be a presence university. So the best is always if we have courses where you are present, I am present, and we can interact. That's always the optimal. And around that, you can always build digital elements. So the Applied Microbiology lecture already had digital elements, for example, these homeworks, which were kind of flipped classroom elements. But now, of course, we have to do everything in digital. But I decided to keep the lecture a live setting, so because that helps you to give you some structure, because you have at each time when you usually would have lecture, you have a lecture, you can join the lecture, and this helps you to keep up with the things and don't watch then the lecture videos at the end of the semester, whenever there will be an exam period. The second thing, it's for me, it is more fun. At least it's some interaction. It is not so boring as sitting at home at the computer and just talking to the computer to have something on the slides. So that's why I do that as a live lecture. So why I do that as a live lecture via YouTube? That's very simple, because it works. YouTube is used to give live streams and so on, and there are different options to do that. There are also pure university options to do that, but most of them are not designed to have 10,000 professors at the same time giving lectures live. And YouTube doesn't care whether you have 10,000 professors more or less, because there is much more traffic on this platform. The chat, as you have seen at present, is also a YouTube chat. I'm not yet completely decided. I have not yet completely decided whether the chat should be YouTube or Moodle. It could be both. The advantage, if it's the YouTube chat, is that you have it open next to your YouTube window anyhow, and therefore it's quite easy to access. The only thing is you need to be registered on YouTube. There was already the question, you can register with whatever name. This might be also an advantage. I don't care what's your name. I want you to participate. That's for me important. And of course, if it would be a Moodle chat, your name would always be displayed there. So you can use your name, you can use whatever you want, but I would be happy if you participate then also in the lecture. Of course, the lecture, the chat should be then restricted to the lecture. Otherwise, I would have to scroll through thousands of smileys and so on to find then the question where I don't have the time for during such a live lecture. Now, live means I'm sitting at home. I'm sitting here in Würzburg, nice weather, flowers all over. But problem is, of course, with my private home internet. And this internet connection, of course, is not a university internet connection. And second, my computer is also not a gaming computer, which is tailored to live streams. So therefore, it could happen that sometimes the picture freezes, and it could also be that the software producing the live stream drops something, and then for half a minute I am gone, but be patient. I'll be back. So I will not leave you before I will tell you now I leave you, and then the lecture is over. So this can happen, but this is something we all have to accept, because of course we cannot buy all professors then now the computers which are thick as you would require for doing live lectures. So this is just the general remark with respect to more the technical aspects of this live lecture, that you know what we do. One thing is also that the live means in this case that it's always a bit of a delay. So the software registers it, then it sends it to YouTube, and then YouTube sends it out. And between the registration and sending it out, you have around about 10 to 15 seconds delay in order to keep the stream stable. So this means if you ask a question, I will see it, don't worry, but it can take some time until you see the response, because this is this delay in between. So that's just for you to have these things clear. Now let's have a look on what we want to do. Obviously it's applied microbiology, so that's why we're here. So I want to start like this, that I first want to explain to you how this module would look like in this semester, and then of course I'm happy to take some questions with respect to the lecture. You can already write down the questions if you want, but maybe some of your questions will be already responded. Maybe you wait a second and then you see what we plan to do now in this semester. Now this is the general information. Okay, it always looks the same. So this is lecture lab course, and the lab course is a block lab course. We will talk about that. This slide you can almost forget now this semester. This is the general structure. Now the readings, unfortunately I cannot, usually I show you the books. Of course I can, because I have these books not here. These books are in, one book I have here, so I have at least the Team and Palladino I have here. So that's nice that I can show at least one book is possible to be shown. Here we are. This is the Team and Palladino Introduction to Biotechnology. However, this is the least suited book for this course, because this is a nice to read textbook about biotechnology. So this is certainly not the number one book here. The number one book here is rather this one, Brock Biology of Microorganisms. So this is the book you should refer to when you go for it, and this is certainly the most suitable book also in this course. Yeah, there are other books, of course. You have Fundamentals of Applied Microbiology, Glaser, Andranikian. This is a German textbook, Angewandte Mikrobiologie. So some aspects of this course come also from these other books, but for the international students, don't worry. So if you have this book and look sometimes into this book, everything is fine, and if you follow the lecture, everything will be fine. Now, so this is what you should learn. Of course, you should learn also something. My mouse today doesn't really work, so maybe I can enhance it by putting here a paper, hopefully, otherwise it's broken. It looks better. So it's the table. Okay, so so this is simply what you should learn after this lecture. And now, I think this is something I don't have to tell you again. If you see this little man, you can lean back and enjoy what I tell you. And this is the general structure of this lecture, and this is already the structure we have in this summer semester 2020. So we have, of course, the lecture, lab course, homework. So these are these three things we have. Lecture, you see here the slides are, of course, on Moodle. I will talk in a couple of seconds about that in more detail. Of course, it's relevant for the exam. I also will talk in a second about the more detailed exam, lab course, and homework. And the grading is like this. The lecture, you will get 100 points from the exam or whatsoever for the lecture. Lab course is just a test state, yet you need that test state to get the complete module. And the homework will give you 5 additional points. So if you have all homeworks, you start with 105 points. How does the homework look like? The homework are 4 original papers which are accompanying the lecture. So these are then sides to the lecture. And then you have self-assessments and Moodle for these papers. You get 5 points if you have done all self-assessments. So of these 4 self-assessments, all of them. 3 points if you have done 3 self-assessments, and obviously 0 points if you have done less than 3 self-assessments. Now, these are real self-assessments. I want you to read the papers and understand the papers. You will see in a couple of seconds, we will talk about treating coronavirus. And you will see how important it is for you. You should become scientists. And the intention, the very nature of a scientist is that he is able to quickly analyze data and critically analyze data. And this is something you have to learn. Yeah, this is not something you learn by looking at slides. This is something by looking on data and interpreting it. And I want to help you with this. And in my opinion, one has to start as early as possible with that. Many others say second semester is too early. I say no, you have to start right away. However, you start in a very mild manner. This is not in the exam. This is a homework. And you can have an original paper. The difficulty level of these papers varies. So some papers are not so easy to read. Some papers are easier to read. And you will have then questions which are tailored on whether you have how to analyze also such a paper, and to understand the charts, and to understand why some data might be weak even though you don't see it right away. And I don't want you to put you under pressure. I want you to do it. So you profit most if you really do it, if you really take these questions also serious and really try to respond them and think of them. But you will get these 5 points if you just have done it. So I don't care whether you then for one self-assessment have responded all responses correct. So you could get for the self-assessment, for example, 10 points. Whether you have 0 points or 10 points, I don't care. I just want you to do it. So that's the important thing, because this is how you learn it. And second, the questions, of course, some kind of these questions are quite difficult. So this is not the type of questions you expect. Not all of the questions in the exam will be like this. That's also the cool thing about the self-assessment. You see where the things go. But you can be relaxed. You just have to do it. If you have done all self-assessment, you will get the 5 points. Yeah? Of course, you profit most if you take it serious, because then you learn. The intention again is learning how to analyze papers. So now we have a special situation. Now we have Corona outside. So the question is, how should this work here? Yeah. So we have this lecture, as you already see now, as an interactive live stream. So with chat, quizzes, and so on YouTube each Thursday. So the lecture is always at the time when we usually would have lectures. I'm aware that this is not all of you might be able to participate. So for people in Asia and so on who sit now in Asia and not in Germany, this shouldn't be a problem. But I'm aware that for people from America, be it North or South America or Latin America, there the time zone is different, and it's of course it's hard at 2 o'clock in the morning if you're East Coast, or I think it's then 6 hours, 2 o'clock in the morning to wake up and join lectures, and nobody expects that from you. So this means that this lecture will be you can retrieve that lecture later on also on Moodle, and the link is on Moodle. Sorry, it was still on YouTube. It's a private link, and this link can be retrieved via Moodle, and then you can watch this lecture whenever you want. So this means that nobody who cannot participate on time has any problems. It's nevertheless, of course, an advantage to be on time, because you have seen the lecture then first, and second, you can participate. So this is also important. You can ask questions and so on. So this makes life easier. Of course, people then from people who are not from let's say a time zone who cannot participate. Honestly, I don't accept if you ask me questions in between. You can participate in the lecture, but of course, people who are from the US or from Brazil or Argentina or whatsoever, you cannot participate in the lecture. Just send me an email if you have questions. That's clear. For all the others, lecture questions which are related to the lecture are to be asked here in the chat. Please do that. That's why we do it, and this is usually because why do I do it in this way? Because usually, if someone has a question, others usually also profit from the response. And of course, I don't want to answer questions 50 times, so it's better to have that here in the lecture. Okay, so the chat opens at 8 a.m. and I will, in the future, there will be at the beginning questions displayed also at 8 a.m. So you can then respond to these questions in the chat, or it depends, it might be that some of these might also be a quiz or so. You can start to put your questions. If you have questions which are open, you can put your questions in the chat, and the lecture then starts at 8:15 like normal. Yeah. And again, it can be retrieved later on. The link will be deposited on Moodle. Now, the homework, as already explained, the self-assessment, this will all be on Moodle. When the homework needs to be done will be announced in the lecture. So this is always then accompanying to the lecture. So the homework is then just, but maybe for the people who watch this lecture later because they cannot participate, it's important that you also watch the lectures in time, because the homework has to be done when I say now this homework until next week. I want you to participate during the complete period and not doing everything at the end, because this will not work. So now the lab course. Of course, a lab course is a lab course, and of course some lab courses, and this is also what we have coordinated, will be done in a kind of a digital manner. However, Applied Microbiology is certainly, for me as a head of the study course, considered as a core lab course. And you need to do something with your hands. And this is, of course, not something which you can learn in the virtual reality. So this means the lab course, we decided this lab course has to be done in a later time point in real. So this lab course will be postponed to 2021. And this will be a block lab course in the semester holidays, either in the semester holidays, in the spring semester holidays, or in the summer semester holidays. We don't know yet. It depends on the situation. But of course, we will keep you updated and we will inform you. Now, the advantage is in your course, that doesn't really matter. So this means in this semester you have more time because there is not this lab course, and therefore you can write. However, the exam, we don't know yet. I will come to us in a second to the exam. And then you do this lab course, you get a test date for the lab course, and as soon as you have passed the lab course, you will get the credits for this course. And of course, you can all follow on courses like whatever, bioprocess engineering and so on. What usually is required that you have to have applied microbiology for this. Of course, you can participate in them, although if you have not yet done the lab course. So that's of course not a problem. So the lab course will be a real lab course, but it will simply be postponed. So it doesn't, for you at present, it doesn't matter. The date when we exactly will do it, we cannot say yet because we have to follow the situation. I'm quite sure that we can do it at the summer semester breaks. We would prefer to do it in the winter semester break, so in February or March. It will be 1 week block per group. So that's the idea at present. Exam, I don't know whether I have another slide. Exam, honestly, we don't know yet how the situation develops. So there will be an exam here for this module. What type of exam, actually, we don't know yet. And of course, as soon as we know the development, we will keep you updated. The exam will be after the semester in whatever form, and for sure the exam will be very, very fair, because the corona situation is a challenge to all of us. It's a challenge for you. It's a challenge for us, and we don't want to. We don't want to punish anyone or so. So I don't want to squeeze you because of this. So in this case, it's kind of a promise. My exams are always easy, but for sure this exam, in whatever form it will be, it's likely to be quite easy. Yeah. Okay. So now let's have a look to the next slide. So this is the overview of what we want to do. Of course, introduction and basics, and then we have all these topics. And due to the actual situation, we will then start with infection biology and vaccines. And of course, we will have a closer look in this semester on coronavirus. So I will talk more than before on how to develop about also the immunology background, which is in here. I will put a bit more focus on how vaccines are developed, and I will put more focus on how this can be tailored against a corona virus, how you can develop vaccines that you can discuss and that you can understand the discussions, what is the best approach and why are these approaches followed. Okay. But now I might have a look first into whether you have questions or not. And so let's see. Oh, there are already many questions. Yeah, so here I have a question. Is it problematic that my name doesn't match? This is, of course, not a problem. So you should, at present, you should ask the questions in the lecture, as I said. So in the lecture, in the YouTube chat. I don't know yet definitely whether we will stay with the YouTube chat. I have already said to you that there are some advantages. The disadvantage is, of course, that the YouTube chat is more open. So if you don't spam it and so on, I would like to keep it with a YouTube chat because it's just for you more easy to use. It's next to it, and for the people who don't want to show their name, I have no problem if you put whatever name. Then it's easier to participate. I want you to participate. I don't want you to blame you if you have a wrong response. So you can keep your correct name. It would be a problem that I don't keep this record and watch if you for whatever exam if you have given once a wrong response and therefore you get a bad grade. If you never get a response, give a response, that's worse because you should participate. Again, the lecture takes a maximum until 10 o'clock. It depends. So it's usually my lectures are more interactive, so therefore the time is also dictated by the interaction, which is then now a bit missing. So the information content, the density of information is higher, and I don't want to have your brains explode by having one lecture where I have the complete content of the complete semester told in one lecture. So it's likely that the lectures will be shorter. So the Moodle chat would be advantage. The question that I would stay and make it easier to rise. So this is not true. This lecture in YouTube is also attached to the lecture, and the Moodle chat is also something which would be a setting where I can put away. I would not restrict. For me, maybe that's a kind of a philosophy how you see a chat. I want this chat to be regarded as a real chat. I don't want to have a chat as a document that everybody looks it up. I will not do it. You should not do it either. Just ask your questions. Don't be shy to ask questions. I will give the examples, the answers usually via the microphone. So I don't write in the chat. I read the questions and I will tell you this or that, and then you know it, and then you note it down. That is the better way, and you hear it in the lecture. And the chat window, as you see, is then displayed, so you can still read the things which are in the chat. So I think that's not a difference between YouTube and Moodle. Yeah, just I can rewatch later as well. So, but by the way, but this can also be both for Moodle and for YouTube, just to make this complete. I can inactivate that. I can both inactivate later on watching of the YouTube chat as well as the Moodle chat. So this is just an easy technical. What about people who did the lab course last year? The exam will be credited for 100 point for them as well. Yes. So that will be the same for the as for the others. Lab courses will virtual. For other lab courses, I cannot tell you. So this question goes to other lab courses. Whether for the virtual lab courses you can repeat the experiments in real later on. I cannot answer. The most likely answer is no. The problem is capacity, because if then all of you then, which I understand the question, which is a good question, but if all of you then redo the lab course because you want to do the experiments, and at the same time you have another 100 students doing the lab course, of course this will lead to a collapse of the system. Self-assessment mean that there will be quizzes on the paper. No. This is a Moodle self-assessment. So this is a Moodle self-assessment. You will see that you will as soon as you have done it, it's quite easy. Yeah. Okay. I think this is it. That the questions now are responded, and then we will go back to the paper. Let's have some coffee that I don't fall asleep during the lecture. It's a problem with the camera on, so that you see that if I'm closing my eyes, that you see it. But also I can talk if I sleep, by the way. So, okay, this is kind of a repeating section because first of all, the introduction, and second, we had some leftover from the microbiology lecture, and I realized that it's a chance indeed that we have this leftover, because we can use this leftover to start to talk a bit about corona and treatment options of coronavirus. Now, let's start with an example what applied microbiology actually means. Now, this is a wastewater plant in Kleve. This is how it looks like. We actually have a project in the student from the upper semester. This is internship in-house, which is an exception, but this is a special case. And he does modeling of this wastewater treatment plant in a software called SuperPro Designer. You will work with this software in the industrial biotech seminar, and he wants then to calculate because they will set up a new production line, and with this model he wants to look whether how to optimize the processes, which is quite challenging and demanding, and we do that together with Umweltbetriebe Stadt Kleve. By the way, just watching this picture, I just realized that we do something here. Now, you have a wastewater treatment plant and a mosquito. I would ask you now, where is the relationship with the mosquito and the wastewater treatment plant? But due to the delay, I cannot always ask you questions, because otherwise I have to drink coffee for half a minute always, and this is also too much coffee for me. So this is the case. So the relation here is, of course, you have in every wastewater treatment plant you have open water ponds, and the problem is that mosquitoes can lay their eggs in these open water ponds. So what about this? A mosquito, of course, has nothing to do with microbiology, but of course, the diseases which are transferred are microbiology, and maybe also the fighting against this mosquito is related to microbiology, and indeed it is. And this is an example of a work published in PLOS ONE, and they have studied the dynamics of two bacterial species, Bacillus thuringiensis and Leucinibacillus sphaericus, and both of these species have a speciality because they form toxins, and these toxins are deposited in the bacterial cells as crystals. And that's of course the cool thing for a microbiologist. You see that in every slide. I think also we have talked in basic microbiology about that, because the phenotype of bacteria is quite boring. They all look the same. And these bacteria have these crystals inside, and therefore you see something under the microscope. And you might remember that these crystals are toxins. So if insects, and these are specific for the certain insects, if insects take it up, they die. Especially larvae, usually they are tailored against larvae. So these are tailored against mosquito larvae. So these are then special strains where this works. And they have analyzed if you put these bacteria in the or spores in this wastewater basins, you want to see whether these then disappear readily, or whether one or the others disappear earlier. And then they have shown it, and they have then plotted this chart where they have just analyzed the kinetics. So I warmly invite you to have a look on that. By the way, people with a not so good internet connection, which see then the slides quite sparely in the future, I will upload the slides before the lecture, so that you have the slides with a good quality, and you can in parallel, maybe with a tablet or so, you can then watch the slides and you see then the pictures better. This will be done in the future. Okay, if I've forgotten to upload it, then you can tell me directly in the chat you have forgotten to upload the slides, and then I will do. Okay, now this is an example of what applied microbiology is, but it's just an example. So this is the pragmatic microorganism list of the definition of applied microbiology, and it's not so easy to define it really. So the basics here is on biochemistry, genetics, metabolism, whatsoever, and applied is then medical microbiology, immunology, agricultural soil microbiology, industrial biotech, aquatic microbiology. And you see that in this course it's kind of a mix. So we talk about metabolism also, which is more part of the basic emphasis. We also talk a bit about infection biology, virology, which is also basics, but which is at the same time infection biology, of course, applied, because this goes in medical microbiology, and this is the emphasis we put here. But we have also courses which are medical biotech, agricultural biotech, and industrial biotech, and to a very large extent they're at the same time applied microbiology, because the microbiology is of course a very, very important feature of biotechnology per se. So it's not, applied microbiology is not just restricted to this course, and this course is also not pure applied microbiology. But at the end, we don't care. It's just the word. It's applied microbiology. Of course, obviously, it's the microbiology which is applied to something, but don't take that too serious with respect to this course. Yeah. Now, I already said that last semester we did not yet talk about antimicrobial agents for in vivo use, and that's what we have to take up this semester. And the good news is that we then can have used that as an example to look for what is happening with Corona. I'm just going from time to time. I'm going back to the chat window. So so just if you have questions, don't feel free to put them in. I'm just looking whether there is a new question coming up. Lab course last year, my lab course self-exam means in lab course of other courses. Yes. Okay. No more questions. Don't worry, I will not forget you. But just if you have a question to a slide, put it in the chat, and then if I see, there is a question, I can go back. The easiest thing is if you then put the slide number, that I know to what slide it refers to, because usually when I see your question, I will be already have proceeded to the next slide. So this slide we have already shown, you have already seen, and you remember. Now, unfortunately, I cannot. I try to look deeply in your eyes. You remember, yes. Huh? all people now at the monitor at the other side, Nick. So, but make the picture bigger. I think then it's more intense. You remember. If not, if now 90% of the students at the monitor on the other side turned red, I didn't see it. This is the bad news about doing with the YouTube chat. I don't see you. That's reality. You see me. I don't see you. So but you need to. This is really basics. You need to remember it. And although you remember it, just to explain it again, we have three basic differences with respect to chemical growth control. We have agents which are bacteriostatic, we have agents which are bactericidal, and we have agents which are bacteriolytic. And the bacteriostatic agents are agents which block growth. So as soon as you apply these agents, the bacteria or the microorganisms can't grow anymore. But they're not killed. That's important. A bactericidal agent kills also the bacteria. So of course it also blocks growth, but at the same time it kills the bacteria or the microorganisms. So the viable cell counts go down. You still see the particles, so it doesn't lyse them, it's just killing them. Yeah? And a bacteriolytic agent both kills and lyses them. So that's the basic difference in between. But important, a bacteriostatic agent only stops growth. In other words, it doesn't really kill them. The bacteria are still there. Okay. We will see at the end, I will show you an example in reality how these subtle differences can break your neck in a real world setting. Yeah. Okay. Now, but we have already talked about chemical growth controls, but we have not yet talked about antimicrobial agents for in vivo use. So you want to treat now a bacterial infection, and we start with bacteria here. The virus section initially was very small, but now I have expanded the virus section due to the actual corona crisis. Now we have synthetic antibacterial drugs. So not all antimicrobial agents are have to be antibiotics coming from streptomyces or actinomycetes. It could also be actinobacteria. It could also be that they're synthesized. So the first antibacterial drugs were indeed sulfur drugs, which were synthetic sulfur drugs. And there are a couple of still in use drugs which are based on nucleic acid analogs, for example, or quinolones. And they are also still in use for treating bacterial infection. For example, ciprofloxacin is still in use, and this is not a drug which is coming from a natural source. So they are synthesized. Yeah. Now, how do they look like, these drugs? Of course, this is something you do not need to learn by heart. That's clear. So we have growth factor analogs. So you have usually they look similar to what the bacteria uses. So you have a phenylalanine, and the analog is this drug, and therefore you disturb protein biosynthesis in this case. Here you will induce mutations. So this uracil, for in this case, of course, not mutations, you inhibit translation, and you have an fluorouracil looks similar to it, and thymine and bromouracil. This will put mutations in your DNA. Of course, you have to use analogs which are recognized by the bacterial enzymes and not by the human enzymes. Otherwise, not all of them, by the way, some of them are also recognized by the human enzymes, and then of course you have quite severe side effects. If you have growth factors which are specific to bacteria, you can do the same, or additional factors like aminobenzoic acid, and then you can use this analog, which is this sulfanylamide, and this then could also be regarded as a folic acid analog. So this is just the basics of these synthetic antimicrobial agents. Make something which looks like but has a different function than the natural ligand. Okay, now let's come to the antibiotics, which are of course the most relevant class today. And antibiotics are agents which were produced by microorganisms, generally fungi and bacteria. Today, most of them are produced by bacteria. Now, less than 1% of the natural antibiotics are useful in clinics. And now my question goes to you. I will check it in the chat. So again, it takes 20 seconds, so I will proceed. Why? Why are just this small amount of antibiotics useful in clinics? So please, question to you, why is this such a small amount? Where is the problem? Now, I proceed. Often you use chemical modifications to enhance the clinical efficacy. In this case, we talk about semi-synthetic antibiotics. And important targets here are of antibiotics are ribosomes, cell wall, DNA, and so on. You can read it yourself. So it's so you attack points. At the principle of an antibiotic, which is useful, is of course attack points which are specific for bacteria, where the bacterial protein differs substantially from the human protein, in order that you have not severe side effects. So this is just an overview of some antimicrobial agents. Of course, I don't expect you to learn that by heart. And you see some antimicrobial agents act on the level of DNA. For example, they block the gyrase. So you remember DNA is supercoiled in the bacteria. It's a circular DNA. Chromosomes are different. Then we have the polymerase, which is attacked here. And many antibiotics interact with a ribosome. You see that a very large amount of antibiotics interacts and interferes with protein synthesis on the level of the ribosome. Some interfere with tRNA synthesis and so on. And of course, the penicillin, where is it? Cell wall here. Penicillins interfere with cell wall synthesis. So you see that this is let's say the zoo of antibiotics which are in use. Now I'm having a look whether you have already some ideas of why antibiotics don't all of them work. Look, so okay, so what is in vivo? You should come to the classes more often. In vivo means, in vitro means this is in your petri dish, and in vivo means this is applied to a living organism. Yeah. Okay. Now, resistance is a problem. Okay, yes, this could be a problem. So there are some antibiotics where you have a high level of natural resistance, but usually this is not the thing that you don't use these antibiotics then in clinics. Most of natural, usually beneficial to the organism, produce them. No. So a definition of an antibiotic, of course, is that you harm other organisms. So for a microorganism, you produce an antibiotic as a microorganism, usually as soon as for example, you're in a certain habitat, and then the nutrients are gone. And then usually you have the problem that next to you other bacteria are sitting and they're consuming your nutrients. So what do? Just kill them. Yeah. And so you then start to produce antibiotics and kill the others. So of course they're harmful for these bacteria. Otherwise they wouldn't be called antibiotics. And in general, bacteria don't kill humans because you don't sit in the soil and lick off their sugar. Yeah. So rarely you do that. Yeah. So the evolutionary intention of these antibiotics is kill other microorganisms. So this is exactly one thing, is severe side effects of antibiotics. So many antibiotics have I said the important thing is that it's a high specificity to the bacterial system. So if you have an antibiotic which is highly specific to the bacterial ribosome, based on there are two effects. There's an on-target effect and an off-target effect. I think this is so important that I put it on. Of course, it's not now for applied microbiology the primary intention, but this is something I wanted you to really hear. On-target effect. effect and an off-target effect. Now, what does on-target effect mean? On-target effect means it's an effect if you have antibiotics which works, for example, on the ribosome. Yeah. So of course, all effects which are due to this are on-target effects. So of course, an effect is that you block protein biosynthesis, and all effects which are then due to that are simply due to this mechanism of action. And of course, if you attack a human ribosome, this is also kind of an on-target effect. So so this is could then, the side effects due to on-target effects are one thing, but you could also have effects on other things, on other targets, and these are then off-target effects. And usually off-target effects are always side effects. So, and whether it is an on or off-target effect, the problem is that many of these antibiotics are quite toxic, and we talk about the therapeutic window. So if you increase the dose more and more, you start to have toxic effects also on humans. And so you need to have a level which is not too toxic for humans, but toxic enough for the bacteria. And sometimes this window, what you have, so this in between, that's the toxicity for which is too high, because then it's too toxic for humans. And this is the concentration you need as a minimum in order to be toxic enough for the bacteria. If it's like this, you have the thing in between which you can use. So this is your therapeutic window. It's fine because your dose needs to be to achieve such a level in the body which is in between. If this is very small, then you cannot really use this antibiotics, because it's impossible to stay in this very thin level. So it's a very highly likely that you are at the too high toxicity level, or that you're at a too low level in order to get a therapeutic effect at all. Now let's see whether we have more responses. Resistance, Kevin said it, severe side effects, severe to general necessary microorganisms in yourself. Yeah, that would be nice if an antibiotics could say, oh, this nice microorganisms in the intestine we keep. Usually they do that, and you cannot really avoid that. So that's maybe, I hope not, but maybe especially the females under you might have already experienced that, or know someone who has experienced that. If you have an antibiotic treatment, it is not uncommon that after this treatment you get a fungal infection at a very delicate part of your body. And the reason is that this antibiotic treatment also kills lactic acid bacteria, which keeps the milieu in an acidic setting. And as soon as they are killed, this acidic milieu goes down, and this then allows fungi to grow there. So that's the problem. So it's rare that you have antibiotics which keep the good bugs alive. So this would be nice, but this is unfortunately rare. Any antibiotics might harm your microbiome? Yes, they do. Maybe how large the damage is. Antibiotics and how they could create superbugs. How do the bacteria develop resistance? Yeah, the resistance development, we come to that, but this is not, the resistance development is not something which impedes the use of the antibiotics. So we had the side effect discussion here, but important is another thing also. Another thing is that antibiotics, if they're useful for treatment, they must come in the body in some cases. So some antibiotics need to be injected. So this is certainly not an antibiotic for daily use. So because injection has to be done by the medical doctor. Yeah? So you want to have an antibiotics which you can apply via pill. And this antibiotics then needs to get into the circulation somehow. This is called bioavailability. And many antibiotics simply don't do that. So you don't get into the circulation. Some antibiotics, you say, okay, this is so important, it has a new mode of action. Then you say, nevertheless, I have to inject it, okay. But sometimes then the antibiotics are too unstable. So if you inject it and in the serum or in the liver, they are readily degraded, so that your serum half-life is very, very short, they're also not useful. Yeah? So if the elimination is too high. Some antibiotics might interfere then very tremendously with enzymes in the liver, which are important for detoxification, and then they can provoke indirectly side effects. And this is, depending also on the antibiotics, then something you would not tolerate. Yeah, if this is a reverse antibiotics, okay, but if this is a broad range antibiotics you want to put in a first line treatment, you wouldn't accept that. So this means a clinical development for antibiotics has to go in more than just in this setting. Yeah, it works against the microorganisms. Okay, so now we go on, and so that's the annual production and use of the antibiotics. And it's interesting, even today, more than 50% are beta-lactam antibiotics. These are the first ones which have been discovered, and they are still the most important one. That's interesting to know. Okay, now what are beta-lactams? So I just pick my coffee again. And before the question comes, of course, you do not have to learn by heart the structure formula of penicillin, but I want you to know what is the basic principle of beta-lactam antibiotics. They are the most relevant group of antibiotics, so therefore you need to understand what happens here. So first of all, the major representatives are penicillin and cephalosporins. Penicillin, this is coming from fungi, this comes from bacteria, but the basic mode of action is in this so-called beta-lactam ring. And what it does is it blocks the cell wall synthesis by blocking the transpeptidase. And this transpeptidase catalyzes cross-linking of two glycans with the peptide chain. You remember we talked about the structure of bacterial cells. So we, both gram-positives and gram-negatives have a peptidoglycan layer. And the peptidoglycan layer is a sugar backbone. And this sugar backbone is cross-linked via short peptides. You also remember maybe that these are also non-canonical amino acids inside, just as a remark. And this cross-link, of course, stabilizes the cell wall. If you don't cross-link it, you just can diffuse away this the cell wall, and then so they're not rigid, and then the bacteria is simply lyse. That's the mode of action here. And this, by the way, this looks kind of like a peptide bond here, but it isn't, and therefore this can covalently block the enzyme which does it. Now, the resistance mechanism, so if you many bacteria are resistant against penicillin, and in this case the resistance mechanism is that an enzyme called beta-lactamase, BLA genes abbreviated, just cleave this beta-lactam ring, and if it's then open, it's no more active. Now the beta-lactams, which are naturally synthesized, are mainly active against gram-positive bacteria. Why? Very simple, because they cannot diffuse through the outer membrane in gram-negative bacteria. Yeah? So they would be active against gram-negatives also, however they don't reach the point where they act. And that's similar to what we have talked before. If you have if you want to apply something orally, which is highly active against bacteria, but it doesn't get into your circulation and body, and the bacteria sit in the brain, it's useless because you need to get it into the brain. And injecting an antibiotics directly into the brain is not something which is suggestible. Okay, now if you then do chemical modifications, especially this group then is modified, you can change the spectrum. And this is on the next slide. Of course, you don't need to learn that by heart, but just to remember. So the natural penicillin, again, this group here, we talk about this group here. this group here now can be modified. So natural penicillin, this NH3 group looks like this, and then we have semi-synthetic penicillins, and maybe the most known one is ampicillin. And you see it's a very simple modification. You have put here an amino group to this methyl group here. That's the only change here. And this now is also active against gram-negative bacteria. This with this simple change. So this is therefore certainly still the most broadly used antibiotics in first-line therapy. Yeah. Now there are other antimicrobial agents for in vivo use, and one group are aminoglycosides. Again, you don't need to learn structure formulas, but you need to know the basic features of this group. They contain amino sugars linked by glycoside linkage, and these usually target ribosomes inhibiting protein synthesis. Examples are streptomycin or kanamycin, as on this picture here, neomycin, gentamicin. Today, the clinical use is moderate because there are many bacteria who have developed a resistance, and the toxicity is quite severe of these antibiotics. So what I talked about is this therapeutic window. You have to go up with a dose, and you usually are in a dose range where you expect already toxic side effects, and therefore usually these are not no more in use. However, we have now re-emerging, for example, tuberculosis settings, especially in the Eastern world, for example, Russia, and many of these antibiotic strains, of these tuberculosis strains, are resistant against a large array of antibiotics, and they have then to go back to kanamycin for treatment. So this antibiotics, which was thought to in Germany, it's even no more approved or it's no more in sale as an injectable. This is now coming back. Okay. Now we come to another group. These are the macrolides. A representative is the erythromycin, and these are broad-spectrum antibiotics targeting ribosomes. Yeah. So then we have tetracycline. Tetracycline is streptomycin, for example, coming from Streptomyces tetracycline, and they're broad-spectrum antibiotics also targeting ribosomes. And these antibiotics they can complex calcium, and then they're inactive because then the uptake is blocked. So if you drink milk and at the same time use this antibiotic, then the uptake is eliminated and the effect is gone. So this means that these antibiotics, you have really to avoid milk at all in principle, or at least I think it's 2 or 3 hours. When you take this antibiotics in the room of 2 or 3 hours, you shouldn't drink milk in parallel. This is not true for all antibiotics, but for tetracycline it's very, very delicate if you do it at the same time.